Comparison of biosorption by live biomass and dead biomass. Biosorption is the process where biomass of plants or microorganisms is used for removal of pollutants like heavy metals or hydrocarbons or azotides from polluted sites including water or soil. Both these processes have their own limitations as well as benefits. So here we are going to compare these two methods. First point of mechanism of metal uptake. Live biomass. The biosorption of metals by live biomass primarily occurs through active metabolic processes including ion exchange, enzymatic reactions and cellular uptake. These methods result in change in the redox potential of the metals. Live organisms can regulate their uptake and secretion of metal ions, allowing them to adapt to environmental changes. This active process may also include the synthesis of metal binding ligands or biofilms to enhance metal removal. Whereas dead biomass, in contrast, relies on passive adsorption mechanism such as surface adsorption, electrostatic interactions, and ion exchange on the functional groups present on the biomass surface. Since in dead biomass metabolic activity is absent, the uptake process is solely dependent on availability and affinity of the binding site on the cell walls or the pores which are present on the surface of these biomass. Adsorption capacity. In live biomass, often the adsorption capacity is dependent on the active metabolic processes which are occurring inside the cell. These processes allow for selective binding and even internalization of the metals and other pollutants, which can result in higher efficiency of pollutant removal, particularly for metals like copper, nickel, cadmium, and azotides, which are having charged groups. Moreover, live microorganisms can adapt to varying metal concentrations over time. The live organisms have the capability to adapt themselves to changing conditions. So even if the pollutant concentration is high in the environment, slowly they can achieve the adaptations and remove the pollutant. Also in the live biomass, we can use genetically modified organism for the same purpose. Whereas dead biomass exhibits lower adsorption capabilities compared to live biomass. Without metabolic processes, the uptake relies solely on the surface interactions between the pollutants and the available functional groups, including carboxyl group, amino group, hydroxyl group, sulfhydryl group. However, it can still effectively remove metals and other pollutants, especially in the systems where high adsorption is not required or when the biomass is optimized for sorption. Live biomass uses active as well as passive ways for adsorption, whereas dead biomass solely depends on passive mechanisms as there is no involvement of ATP molecules. Regeneration and reusability. Live biomass. Regeneration of live biomass is more complex as it may require specific environmental conditions like presence of nutrients, temperature and optimum pH for recovery. Furthermore, the metabolic processes may be disrupted after multiple cycles if the same biomass is reused again and again. This reduces the overall longevity of the biosolvent. Additionally, long-term exposure to toxic metals may lead to the death of the biomass, diminishing its effectiveness. Whereas dead biomass is generally more easily regenerated as the absence of metabolic activity allows for straightforward desorption using mild chemical agents. So when we want to remove the pollutant from the biosorbent surface, different processes are used where alkalis or acids are used or sometimes high temperatures, electric currents are applied. So there are always chances of disruption of live biomass, whereas dead biomass can easily treated by using all these processes. Moreover, since dead biomass is not alive, It is less sensitive to toxicity, making it potentially more durable over multiple cycles. However, the biomass can degrade over time due to physical or chemical wear and tear, especially in harsh operational conditions. Next point of comparison is operational simplicity and cost. Using live biomass in biosorption processes is often more complex and it may require careful control of environmental conditions, including oxygen supply, nutrient availability, pH, and temperature. Also, when different organisms are used together, there are always chances of competition. So, all these factors need to be monitored properly so as to achieve maximum efficacy of the process. These additional requirements in live biomass increase operational complexity as well as cost. Furthermore, live biomass may require periodic replenishment, adding further cost to the process. Whereas in dead biomass, the use is simpler and offer more cost effective as compared to live biomass as it does not require the maintenance of metabolic processes or growth conditions. Its use is operationally straightforward and it can be sourced from waste materials like agricultural residue, fungal biomass, or spent yeast, which makes it an attractive choice for large scale application due to its lower cost. Environmental and sustainability considerations. While live biomass is more efficient in removing metals and other pollutants, its sustainability may be limited by the need for specific environmental conditions and the risk of biomass toxicity over time. Additionally, maintaining live organisms could have a larger ecological footprint due to the necessary resources like nutrients and energy for sustaining growth and activity. There are always chances of unintended growth of these organisms as they are rapidly proliferating in the controlled conditions. In case of dead biomass, the increase or proliferation of the organisms is not considered because dead biomass has lost all its capacity of multiplication. Dead biomass has a clear environmental advantage since it typically comes from waste streams. It is often more sustainable as it does not require additional resources to maintain biological activity and can be disposed of or regenerated after use easily as compared to live biomass. Moreover, utilizing waste biomass can reduce landfill waste and contribute to a circular economy. It works on the principle of waste out of waste. Selectivity and adaptability. Live biomass. One of the primary advantages of live biomass is its adaptability and selectivity. Live organisms have the capability to adapt themselves to changing concentration of the pollutants and surrounding conditions, and they may exhibit preferential uptake for specific metal ions or any particular dye or hydrocarbon depending on its metabolic pathways. This selective binding can be beneficial when targeting specific contaminants such as toxic metals in industrial effluents, whereas dead biomass do not have the capability of adapting themselves to changing conditions. Dead biomass is also less selective as compared to live biomass as its uptake mechanisms are passive and they are mainly based on physical and chemical interactions rather than metabolic processes. While it can absorb a broad range of metals and other pollutants, its lack of adaptability to environmental changes limits its selectivity compared to live biomass. Potential for toxicity or bioaccumulation. Live biomass may lead to bioaccumulation of metals and other pollutants, which could pose environmental risk if the biomass is disposed improperly. Additionally, some metals could disrupt cellular functions, leading to toxicity in the biomass and reduced biosorption capacity. Disposal of biomass containing high metal concentrations also requires careful handling to avoid contamination. Whereas in uh, dead biomass is devoid of all these factors. It avoids the issues of bioaccumulation since it is no longer metabolically active. While it can absorb and retain metals, does not allow the internalization of metals or other pollutants. That means other pollutants do not enter the cells of these organisms, making it safer for disposal. However, like live biomass, spent biosorbents still need to be treated as hazardous waste in some cases, especially when the heavy metals are involved. So, in conclusion, we can say that both live and dead biomass have their merits as well as demerits in the process of biosorption. Live biomass excels in terms of higher metal uptake capacity, adaptability, and selectivity, but it demands more complex operational conditions and poses greater risks of toxicity and bioaccumulation. It also requires more uh, cost and the conditions like nutrients and surrounding conditions need to be maintained. In contrast, dead biomass is simpler, more cost effective and sustainable, although it typically has a less absorption capacity and lacks the selective advantage of live organisms. The choice between live and dead biomass largely depends on the specific application, desired efficiency, and operational constraints.